Welcome, everybody. Most importantly, I know a lot of people have traveled very, very far to be here, so welcome. It's so good to have you here. To all of you out there online, thank you so much for tuning in. Please tell us in the chat if there are times that you can't hear or something is weird, and we will try to fix it as soon as we possibly can. All right, so I just want to, I like to start every meeting like this. Hui has recently partnered with the Wampanoag tribes to develop a land acknowledgement, and so we like to start our meetings this way. We are gathering here today on the traditional unceded lands of the Wampanoag, and I'm just going to put this here and give you a moment to look it over. Think about where you have come from and the indigenous peoples in your area, and that includes all of the people in the Zoom. If you want to add that to the chat where you're calling in from, we totally welcome that. The next thing we like to start our meetings with is the Code of Conduct. And for this meeting, we'll be using the Ocean Carbon and Biogeochemistry, or OCB, Code of Conduct. I'll tell you a tiny bit more about OCB in a minute. Most importantly, just be nice, be respectful, and be a good listener. We're all working together on a really hard task over the next 3.5 days. You will see these QR codes posted up around the meeting spaces, where if you have an experience or you witness something disturbing, just click on or scan the code into your phone and you can report it. You can read our Code of Conduct in detail there, or come talk to me, or come talk to May, and we'll take care of it. It will be handled immediately. But please, really, I don't need to read through this. You're all humans. I see you all hugging each other. You've been colleagues for a long time. Just listen. Take time to listen to each other. One of the biggest parts of the Code of Conduct for this particular meeting is that we ask our speakers, including myself, to please stay on time. We've got a lot to do here. So, you know, we've got an agenda and it is packed. We're going to have a lot of small group interaction time. And for those of you who are leading those breakouts, please make sure that everyone has a voice in those conversations. A small group of 12 to 16 people may seem unintimidating to some, but for others it might not be. So please just be mindful of that as the breakout leaders especially, and make sure that you're making space for everybody to have a voice in the conversation. Just a very small bit, I think everybody here knows what OCB is. If you want to know more, go to the website or come talk to May and me, because we're immersed in it all the time. The Ocean Carbon and Biogeochemistry Program and Project Office have been around since 2006. I came on board in 2007 after finishing a postdoc here, and never imagined I'd be at Hui for this long, but I started working with Scott Doney, that was a long time ago, and we have since built a legacy here with the community that we have as part of our network, and the fostering of activities that have supported biogeoscapes have been a really big part of that legacy. You guys have done a lot of hard work and championing to make this happen. And so I hope that you'll just take some time to celebrate that while you're here this week, because that has been a huge task. And I know a lot of people here who have worked really, really hard to accomplish it. So OCB has these 5 overarching areas. OCB does not have a static science plan. OCB is a very responsive, adaptive, bottom-up program. You can see a list of our current research questions on our website, but really it's the guardrails are basically these 5 icons up here in green. If you want to learn more, like I said, come talk to me or go to our website. We are on Twitter, and I refuse to call it the other name, so the bird will stay on all of my slides for the rest of eternity. Follow us on Twitter. We actually co-tag a lot with Biogeoscape, so you can learn a lot more as well. Just to give you a sense of all of the activities that have come to bear to make this a reality, these are only the ones OCB's been involved with, but you know, the original scoping workshop that kind of raised this idea of bringing together the molecular biology and the chemistry measurements to really understand the microbial underpinnings of the biogeochemical cycles of the ocean and how they're changing in response to climate-driven changes. This was a scoping workshop in 2010. I believe Eric Webb is in the room. I believe Jim Moffitt is online. I'm not sure if these other champions are here or could be here with us. I know some of them had to be out of town for another meeting this week, but this started a long time ago, and it's amazing to me how far it has come. The GeoMix cruise, so many of you are here in the room that participated in either the cruise or the post-cruise workshop from that, which was really an initial pilot activity for this. That happened. That was in 2012, 2013. And again, championed by you. And there have been a lot of developments over time since then. Largely, I think the cruise, that pilot cruise, gave people a sense of what it really takes logistically, the interdisciplinary interaction and the planning for how to do these measurements and sample collection side by side. It's not trivial. And so I think that cruise really gave people a sense of what it will take to build something like this on a much larger scale. There was a small meeting here in Woods Hole in 2018 that was big, an international team, kind of like a small version of what we have here today. And that was focused on putting some guardrails and some sense of foundation and structure, or a vision, a preliminary vision for what this might look like on a global scale, a big international program. And that small group, I think most of those people are in the room today and can tell you that's where the BioGeoscapes name came from. And that meeting really, it catalyzed all of these national activities. You can see now 19 national reports on the BioGeoscapes website. That's amazing to me. 19 reports, 19 national meetings. There are enough people in all of these countries to develop the momentum to build a big collaborative program like this. So that really kicked off a bunch of national meetings all over the world, and OCB helped host the one in the US, which had to be virtual because of COVID, in November of 2021, so 2 years ago. And a lot of the things that have been identified over time here, one of the big ones is intercalibration. You know, GeoTraces has set the bar pretty high for how to do this right, how to do this well, how to do this thoroughly, how to dedicate time and energy to it. And I think that has set a very high standard for us to think about, and we've convened a few activities around this, and I know more are coming. But really getting the molecular, the omics piece standardized, intercalibrated, working together to do intercomparison exercises. So you can see on the bottom here, there are two intercomparison and intercalibration activities that have happened. The reports from those are available on the website. And then more recently, of course, the development of this ExcelNet, this NSF funding opportunity that gives us the opportunity to bring together an international network of networks, to get all of the players in the room who need to be involved to make this vision a reality. And I know you can't read all of these little bubbles too well up here on the screen, but it's incredible to me that all of these little bubbles actually exist, and that so many of the people from these bubbles are here in the room. So OCB's been involved since the early stages of this, and it has been a real honor to be part of it, to watch this community grow, to watch the grassroots hard work and collaboration that's been required to drive it forward. I'll say that Mac is going to be giving a lot more detail about what I just presented there in the brief history of time. So you'll hear a lot more about that. But please talk to some of the people if you're new to this game and you're here and you're like, I don't really know quite what this is, but I'm really excited about this meeting. Come talk to some of us and learn a little bit more about the history and about all of the planning and all of the championing that's been involved here. Workshop logistics The Wi-Fi is up on the whiteboard, okay? And that's the first thing everybody asks when they come to a meeting. And it's there, and I want you all to be able to have a tether to your lives at home. But most importantly, I want you all to be present. Because we have a lot of hard work to do today to distill the key elements of a science plan that people all over the world can plug into to achieve our vision. The participant documents folder, if anybody is having a hard time accessing any of the workshop documents, please come talk to me. I have sent that link out several times, and I want to make sure you all can get there because the agenda I was literally updating the agenda yesterday, so things are in flux here. This is a challenging meeting. There's a lot of challenges associated with the work that we have to do together. So if you need any help, just let me know. Most importantly, the restrooms are behind the auditorium. We're going to give you breakfast and lunch each day. There'll be breaks. The caffeine will be flowing. And if there isn't caffeine out there and you need it, I have coffee. I have an espresso machine and tea in my office. It's downstairs, one floor. I will hook you up. We're going to have a welcome reception tonight. Any of you who have brought posters, please get them up on the boards. I know we don't have a ton of posters, but we have posters, and I want those people to feel the fruits of their labor were worthwhile. So please go and talk to them and check out the few posters that we have, mingle with each other. I want to remind you of the rules of hybrid. Speak clearly. If you're asking a question out here in the audience or online, please say your name and where you're from. Please wait for the microphones. For those of you folks who are here in the room, wait for the mics to come to you so that the people online can hear your question. And for those of you who are online, please wait until you are unmuted or make sure we can hear you before you start asking your question. We're going to do a lot of breakout work. There's a lot of breakout work. Setting up all these breakouts was a nightmare, but I think anyone who asked for a leadership assignment or a note-taker assignment has gotten one. So please make sure, like I said, go into the participant documents folder to see them online, or I'm going to print out the daily day of the agenda and the daily breakouts. They'll just be one day at a time up there on the wall, so you can look at them on paper if that's better for you. They are assigned, though. And for those of you who didn't fill out the Google form, I just assigned you. So be a good sport and go to your breakout. We're going to have a period at the end of, so each of the science themes, we're starting with theme 1 today, and I think we're going to do half of theme 2. Each of the science themes has 2 sessions devoted to it. So you'll note that we have 6 subtopics under each of the science themes, so you'll get to visit 2 of those during your breakout sessions. You'll spend an hour in each breakout session. And then we will have time, we're going to actually have people, in addition to Google Note taking, we're going to have you develop a big sheet of paper. For those of you who are online, that will look like a Jamboard. For those of you who are here in person, we're going to be distributing those big, huge easel pads. And I want you to distill your conversation in that hour onto one sheet of paper and tack it up on a board, tack it up on a window, tack it up somewhere. And when it's time for cross-fertilization, those of you who are here are going to go around with your sticky notes, and you get to see what happened in all of the breakouts. And you can add comments to it or questions or ideas, whatever you want to do. So that's what cross-fertilization is about. And then we will have a period of synthesis. And that's where we'll have one representative from each theme topic, subtopic, or implementation topic. We'll go into that back room and spend an hour distilling it into 1 or 2 or 3 or 4 slides. You'll see the instructions. Anything that's on the agenda in orange, those are instructions for you to follow. We'll remind you about the breakouts when it's time. But please, think big. We're developing a science plan for collective benefit here. So please maintain the focus on this big picture during the breakout and plenary time. If you want to talk about your research conversations or get into the weeds, we have lots of informal interaction time for that. We've got receptions and food and lots of time for that kind of interaction. But please, when we're in workshop mode, Please put your head at that higher level. Think science plan. Think about people around the world plugging into it. There are lots of opportunities to gather with each other. We're going to have the small group work. Make sure that you maximize your interaction. I've tried to mix you all up in as many ways as I can. Try to talk to different people every day. Sit with different people every day. But really, you can be developing new collaborations yourself during this time. There's an opportunity in the folder to set up gatherings. Let's go out for drinks at Aquatic Brewing on Tuesday evening. Tuesday evening's free for all of you, so start thinking now about what you want to do and put something in that spreadsheet and plan. But just a reminder, there are 72 people in this room. Over 90 people as of yesterday had registered for the Zoom, and hundreds have contributed to this process through the development of 19 meeting reports, right? There are only 72 of you here, so you have really important work to do, so keep your eye on the prize. And for those of you online, we need your help too. So be part of this. Please don't check out during the breakouts. We want you here in the breakouts. You're not assigned though. For those of you who are online, you get to choose your own breakouts when the time comes. So we'll set up the rooms and you get to just join the one that you want. This is just going to be for you to be keeping your eye on the national meeting reports and all of the programs that have come before us. That is what has led us to this moment. We're here together. We want to develop something big. We want field efforts, we want lab efforts, and new collaborations and new funded research to come out of this. So this is the prize. You're going to see this slide a lot during the meeting. I will put it up to inspire you as often as needed. I want to just have our organizers and ExcelNet PIs please stand up just briefly. Their names are listed here on the slide. Give them a huge hand because this has been a lot of work. Finally, you're going to see something that looks like this. We need champions. The ExcelNet has a lot of components to it: modeling and data integration, standardization and intercal, informatics and data management, and education and capacity building. And if you want to contribute to any of these, scan the code and sign up. We need your help. We don't want to just do this ourselves in a vacuum. We want community engagement as part of this. So please do sign up. Come talk to me if you can't get to the Google form. And that is all I have to say. I've already broken my own rule by 2 minutes. So thank you.
Too early.
Hello, everyone. I'm Max Saito. I'm here at Woods Hole, and I'm one of the ExcelNet PIs. And I'm really excited to welcome you all here. It's super exciting to see all these faces, people I know and people I don't know from all over the world convening here in Woods Hole to try and help with this effort to build this BioGeoScapes program. So I wanted to welcome you all and just express how excited I am. This is really a presentation on why we're here and how do we get here. And I want to acknowledge that I'm representing this large community of people who've contributed over the last more than 10 years to try and build this program. I really want to acknowledge all those people's efforts. Yeah. Okay. And thank them for their efforts, and just say I'm super excited about this week. There's a lot of young scientists here, a lot of young energy, and I want to encourage those people to engage as well. This is really a program that's for the next generation of scientists, I think. So to set the stage, I want to talk about some of the science motivation for this. And as we all know, there are really large changes happening in the world right now due to anthropogenic influences. There are major changes in global and regional biogeochemical cycles. Here are some examples from the nitrogen cycle and oxygen losses in the oceans, as well as increased iron inputs. And these are example papers on these topics. So really profound changes, and these are just 3 elements, but we know that there are many elements that are being heavily impacted by human activities. Carbon, of course, one of the main ones. There are also major impacts that are happening on fishing, on climate, and through chemical inputs that are affecting marine life. And this is a nice review paper by Carlos Duarte that points out the influence of overfishing as well as deforestation and habitat loss, and how chemicals and fertilizers as well as climate change are all impacting the marine life on different timescales and with different intensity, but there's an opportunity to acknowledge this and try and rebuild marine life. And of course, if we go to the poles, there's really profound effects that are happening at the poles in the cryosphere at both poles. And just this last summer, we of course had record low Antarctic sea ice in Antarctica, and this is something that was picked up by the popular press as well, so it's well in the public's mind. All of these effects are happening, very large effects on the ocean environment, and underlying all of this are microbes and biogeochemical cycles, and this community knows well that microbes are really the engines that catalyze a lot of the key biogeochemical cycles, and understanding them and their influence is really fundamental to understanding Earth's life support system. So there's a lot of basic science there that we need to acknowledge and work hard on to really understand how our planet works and then how we're affecting it, to understand those human perturbations that are happening that I talked about before. And our colleague Al Taliabu has an opinion piece coming out, just came out today in Nature, describing some of the challenges of understanding biology and microbes in climate modeling, and sort of emphasizes this, and he'll talk about this a little bit more this week. But he acknowledges directly the importance of a program like Biogeoscapes, names it directly in this Nature piece that came out today, has a key way to move this science forward to try and help our ability to climate forecast. So if we step back, one can realize that we actually don't even know where all the microbes are in the oceans yet. And our colleague Alison Santoro wrote this really nice opinion piece, a crystal ball piece in Environmental Microbiology Reports, where she pointed out that she would like to see a microbial map of the oceans, and that we don't have this yet. We have this in small parts of the oceans, and particularly in the upper water column of the oceans, but full depth understanding, we still lack actually where the microbes are in the oceans. And she points out that this great quote at the end of her piece, talking about how throughout history maps have been used to shape the world, to shape opinions, wield power, and understand the world. And a microbial map will be no different. So first order principles of just knowing where the microbes are is a goal that remains to be obtained. And in recent years, there have been new technologies that have really come of age that can help this, in particular on the omics and the micronutrients fronts. As we know, there's been a real revolution in omics sciences. This includes the genomic, the transcriptomic, proteomic, metabolomic, and lipidomic, and perhaps other omics measurements. And these capabilities are really astounding in the depth of the data and the kinds of data that are possible. And a great example of this is the TARA program and Atlantico programs that have really pioneered high throughput measurements all over the world and open data efforts. In parallel with this, the ability to measure trace elements and micronutrients in the oceans has really come a great way thanks to the GeoTraces program. This has been through enhances of measuring really low quantities of metals that are important to life in the oceans. This has brought us a lot of new knowledge in terms of how those metals can influence biology. And this is just an example of a compilation of ocean sections where each one of these is a different ocean expedition where they found hydrothermal plumes of iron at the vents for the first time, and huge multi-thousand kilometer plumes, as well as coastal inputs and scarcity in some regions. And so this has really been transformative. This is, prior to the 80s and mid 70s, there were really no good trace metal measurements in the ocean. So we've come a really long way very quickly. But there's actually very few studies that combine these kinds of omics measurements and trace metal measurements, and that's something that also remains a goal to be obtained. And to talk a little bit more about the omic capabilities, I think it's worth focusing on that here and the different kinds of that. We can detect what organisms are present, from viruses to archaea to bacteria to eukaryotes, from microbes to protists, and that allows us to study complex coastal and dynamic ecosystems. But also, in just the past few years, the ability to look at higher organisms in metazoa has really come a long way with eDNA. And now we can, scientists are scaling to see fish and other populations simultaneously on samples that are very similar to what people are collecting from the microbial populations. And that's really exciting because it connections to fisheries and as this sort of holy grail of connecting from microbes to higher trophic levels. And those are all nucleic acid-based measurements shown here on the top. But there's also the capability to assess functions through transcriptomics and proteomics. And that is really exciting because then we can start connecting to what processes are happening and how organisms are responding. What stresses are organisms being impacted by, and what key enzymes may be present, and can we measure them directly to help inform our climate models. And also we can detect metabolism and carbon cycling directly with metabolomics and lipidomics. And that's an exciting area that's really been developing fast. The organic geochemists have really made some incredible progress in recent years. And so there are some big challenges of creating ocean programs, such as TARA or GeoTraces or other programs that come before. And that means we have to scale we have to have bridge multiple scales and disciplines. And we have to redesign our physical and chemical sampling programs to match biology. We'll need to establish standardized sampling and analytical protocols. And there'll be a lot of breaking of boundaries, cultural boundaries between oceanographic communities and omics communities. We'll need more reference genomes and more data to be developed for that process as well. And then there's the data integration challenge, right? These are big data problems. The omics in particular have a lot of data. We thought GeoTraces was a lot of data, but omics will be a lot of data. And then connecting that with laboratory studies, physiology, and then creating new tools to assess this, be it models and improving rate measurements and so on. And then last but most importantly is training the next generation of scientists to do this kind of interdisciplinary work, right? And that's what is really exciting about the Bioscapes community before, because I think we're already seeing it in the abstracts that are being submitted. The young scientists are really excited about really trying to connect across disciplines. And that's what we really need to move this forward. And so, as we know, studying oceans is very complicated, right? We have complex food webs and interactions, and we have underlaying that these biogeochemical cycles. There are physical processes, biological, chemical, and geological processes of various scales that are happening. And this is a 4D problem. We don't just have the space problem, but we also have the time dimension as well. That adds that fourth dimension. And as we know, biological communities can be dynamic and seasonal and have patchiness. So that adds a real challenge to trying to really embrace the biological processes. So we'll need more data, we'll need process studies, observations, surveys, and time series to scale to this. But there are also new tools that are emerging that can help scale to address this complexity, such as bioinformatics and artificial intelligence and modeling and autonomous sensors and platforms. So as Heather mentioned, we've been really trying to build this effort for more than a decade. And here's a timeline we put together of some of the events. Apologies, I'm sure we didn't include everything, but it does go back to that 2010 molecular biology of biogeochemistry workshop that happened in LA, and where some of these ideas of trying to add a much more biological component to some of the GeoTraces efforts were really initiated. And then since then there were, the BioGeoTraces efforts and geomics efforts, what we're calling proto-BioGeoscapes types cruises were launched where people were trying to combine these efforts. And in 2016, there was an OCB and GeoTraces synthesis workshop where people also started really exploring these ideas. And then in 2018, we had this small meeting here bringing the community together to try and assess international interest to try and move forward. And then we had this series of national meetings that have happened. And then in 2020, just 2 years ago, we had a Royal Society meeting where the topic of biogeoscapes was brought up and discussed in depth at the end of that meeting. And then the EU community organized a pan-EU Croatia meeting in 2020 as well. And then just more recently, we had this AccelNet grant funded that allowed us to sort of formalize these efforts and start trying to move this forward. Simultaneously, the DOM Intercal group started moving forward. And here we are today in 2023 at a science planning meeting, and just this year also a meta-transcriptomic community got organized. And then these other events are in the future, a modeling meeting, an informatics and data meeting, and intercalibration assessment meeting, with the hope of trying to launch something in 2026 or so, roughly mid-decade. So this 2018 meeting was our formative meeting where we really brought together people from 10 nations who we knew were interested in this effort and said, okay, do you think we're up for this? Are we ready to try and move forward? And if so, can we write a short document that crystallizes this and come up with a name and send people back to their nations and to the international community and try and garner interest in their countries? So this was just happened just down the street here at the National Academies Institute. And it was a really productive meeting. that helped really set the stage for everything that we're seeing here. And from that meeting, we tried to crystallize this effort and came up with the BioGeoscapes name. And at the core of this was this concept of ocean metabolism being at the heart of our planetary life support system. And we recognized that there were intercellular processes that then were really fundamental, connecting to organisms and assemblages, but then scaled all the way to these ecosystem and global scale feedbacks. And we wanted to develop this kind of effort so that we could constrain the biological and chemical feedbacks on a changing planet. And really asking the question, how do microbial food webs shape biogeochemical cycles and respond to and influence global climate change? So this vision really is one that's trying to integrate approaches from omics and chemical measurements, conducting a global scale quantification of microbial communities, and building a better understanding of ocean metabolism and its influence on ecosystem health and biogeochemical cycles. We recognize that no one country has enough resources to do this alone, so we want to work together and combine efforts to do this as an international community. It's very much a bottom-up effort. There's no one, agency or institute that's saying, all of you need to go do this. I think as a collection of scientists, over the years, many of us have been really compelled by this science effort. And there's hopes to create full-depth basin scale ocean sections, as well as transects, time series. A lot of countries express strong interest in time series, and we'll hear more about that from Maite shortly. Process studies and laboratory studies to try and pull the data we need to do this. And this would result in rich biological and chemical data sets with improved integration between observation and models. And here's a graphic that Naomi Levine and Lilith produced to describe this scale of in situ observations and omic and geochemical data sets and data processing that will happen, and then connecting to models. So in order to do this, we'll have to connect to existing infrastructure and technologies and partnerships. And that involves this connecting to the trace metal micronutrients and omics community in the ocean environments. And these are really active fields, but bringing them together in earnest now. We'll need real substantial intercalibration and data science efforts, right? And I think that's one point I think we want to keep hitting home, that underlying this effort, we really need some really important work to get done to make this work. And that's because we want to collect data from around the world, and we want to be able to compare it from around the world, right? We want a line that European nation conducts to be able to connect to an American line and be able to connect those data sets and interpret them, right? And that requires intercalibration and data standards to do that, right? And then there are also infrastructures that will include multiple sampling modes, trace metal rosettes and submersible filtering systems, autonomous underwater vehicles and samplers, sequencers and mass spectrometers, and so on. And here are two examples of vehicles that could help do this. And of course, Cleo was downstairs right on the doorstep. That was kind of cool. They put it right there. So what is BioGeoscapes? This is a question we get asked very frequently. And of course, this community is really defining it. But I think, from what we've heard so far in our previous meetings and from the national reports, and so what's bubbling up to us is that it's really a program that will be interdisciplinary by definition. It will combine some aspect of omics and physiology and biogeochemistry and modeling. Hopefully, a lot of studies are going out and doing one of these things, but by definition, we want people to try and combine these, right? And go beyond your comfort zone and then try and bridge that gap for interdisciplinary knowledge and extend what's possible. Can we connect micronutrients to the microbial ecology and really dig in deep to understanding what processes are happening, for example, and the organic geochemistry of the metabolites that are moving around, and then connect all of those to the modeling and ground truth it and physiology studies in the labs. And so because of that, it really needs to be rooted in intercalibration and data standards for us to connect the dots. And so to get this going, we were fortunate to have efforts to try and procure funding to get this moving. And there were 3 efforts that had been launched, and we were very lucky in the US to get this AccelNet funded. And AccelNet is short for Acceleration of Networks. It's an NSF program that's specifically targeted to fund international collaborations. And so it was entitled Development of International Network for the Study of Ocean Metabolism and Nutrient Cycles. It came very much out of that 2018 meeting, and the list of PIs and international collaborators is shown here. And of course, this network is already much larger through the organizing committee for this, and hopefully some of you, or many of you, will be involved later on in these future meetings as well. It's important to point out that while this is a generous grant, it actually doesn't fund any science directly. It's purely for organizational and really for what they're calling the acceleration of networks, that bubble diagram. We're trying to build these communities and get them talking to each other and strengthen those networks. And that's why this meeting in person is so important. The coffee breaks are so important. We want people to get to know each other. If you don't know someone, please go out and introduce yourself. We want relationships to be built across international boundaries so that collaborations can be forged so that this program can be launched, because those personal connections are really the foundation for this. Our timeline of activities is shown here. This is what's in the proposal, and we're actually doing pretty good with maintaining this schedule. You can see we are here at this science meeting, and the hope is in the next 2 to 3 years to launch an informatics and data meeting, to launch an intercalibration review and assessment meeting, and to launch a modeling and integration meeting where we bring modelers and observations together to try and to really develop that component as well. We also have a bunch of educational opportunities as well, and I'll briefly touch on that, and you'll hear more about that in the future. And so there's a lot of momentum that's been building for this BioGeoScapes effort over the years. You know, we have national ambassadors for 25 nations so far that have gone back to their countries and organized efforts. That we've also had national meetings from and workshops that have happened. And we've got a lot of interest. We've got, I think it's actually up to 64 countries that have been looking at our website that we know are thinking about BioGeoScapes. And we really want to emphasize that this is an open and inclusive process. You know, we want people to come to this program and try and find a place in it and work with us to try and do some exciting and new science. So that's really a really key point that we want you to take away, people of any career stage and from any nations. And to date, we've had more than 25 ambassadors selected from nations. And I just updated these today. We have over 500 newsletter subscribers, over 1,100 formerly Twitter subscribers, and more than 3,000 users from 64 countries that have hit our website. And we've had, I think we can also see the impact from the ocean sciences. We've run several ocean sciences sessions, and there's just a lot of interest in applying to the sessions. We're seeing a lot of great interdisciplinary talks there and posters. And so I think that also shows that the community is very interested in this effort. You know, we're not having to find the community is embracing this. So key to this effort is really launching these, is this intercalibration and data standards efforts. And I want to give a few highlights from intercalibration efforts. The Ocean Metaproteome community, which I'm a part of, has been launching an intercalibration effort since 2018 with OCB support. And the community is shown here, and there's a recent manuscript that was submitted. The nucleic acid community, as you heard about, has done a heroic effort in writing this really comprehensive report that outlines a roadmap towards community-wide intercalibration and standardization, and that's available online as well. There's also since then been a metatranscriptomics intercalibration that was just launched by Harriet Alexander and Natalie Cohen as well, a small one in there. I think they're hoping to follow that up as well. And then the organic geochemistry community is really interested in these kinds of new programs as well, and they've been very active in exploring intercalibration and comparison efforts. Here are two efforts that are, and the one in the middle is the one that's most recent, this ongoing intercomparison of liquid chromatography, tandem mass spectrometry data, dissolved organic matter analysis data sets. And very recently there was a labile DOM workshop co-hosted by OCB and CCOMP, the Center for Chemical Currencies on a Microbial Planet in University of Georgia. And they had a session directly on trying to address these methodological challenges. Your next intercalibration could go here. You know, I think a lot of people say, well, what parameters will be in BioGeoScapes? And I think at the core, we need parameters that are intercalibrated, right? We need data sets that are intercomparable, and that requires intercalibration. So if you think there's a parameter that could be useful in this program, I think, you know, organize your community to try and go out and organize an intercalibration to show the data readiness and reproducibility, and also to encourage more people to learn these methods. One thing that people don't realize that we all learned in GeoTraces is that when you run an intercalibration, it can seem kind of scary at first, like, oh, you know, we're going to submit our data to someone and they're going to judge us, and that sounds scary. And it is, you know, I'll acknowledge that. But there's incredible value in it. Like, you realize you send your data in, and a few people, you know, maybe outliers, or there's a spread, and then you all come together and like, what happened? And like, let's figure it out. And together you work together, and then together you gain confidence in these numbers, and then you can go. And then suddenly, you know, people who might have been your competitor before are your colleagues that you work together on, and they're your collaborators, and you trust their data. And the community actually becomes much more connected. So that was something we realized. And so you know, I encourage people to not be scared of intercalibration and realize there's really some really positive attributes to it. Also, a lot of people learn methods along the way in intercalibration, right? You can participate when you're still new to a method, and then you come out of it as basically an expert, right? So I think there's some real important advantages to intercalibration that outweigh the scariness of it. So this program has also been launching some webinars, and Harriet Alexander and I have been spearheading this effort. We'd love more volunteers to help this. We've had some on intercalibration and some on data management and on informatics, and there'll be more coming up. These are ways to sort of organize the community, get us talking and thinking about the challenges, so that when we get to those in-person meetings, we're more productive. So these are really useful. There's a lot to think about in this space, in these challenges, and there's more opportunities for webinars as well for different topics. So please feel free to engage on that. Most recently, we launched a BioGeoScapes Fellows program. This is a means to try and embrace the energy of early career researchers, to try and help them connect internationally. We were really pleased to get nearly 100 applicants for this effort from around the world, and we're planning to try and do another round of applications next year. There were so many great applications, we couldn't accept them all, but we were really excited with this cohort that was funded. This provides travel support for international collaboration to help support the BioGeoScapes program, and we're encouraging them to think of ways to help this program as well. So welcome to these new fellows, and we're excited to see where this goes. And so in the last few minutes, I wanted to sort of talk about the GeoTraces Science Plan as an example. There's many global programs that have science plans, but this is one that many of us were involved in, and I think it's a good model for what we're trying to do here. There was an international workshop in 2003 in France, in Toulouse, and that was based on regional planning workshops, and over 300 individuals that participated in meetings and submitted written data to produce this document. It's a document that has a number of chapters shown here, introduction, and then various science themes, 3 science themes, and then it has shorter chapters on synthesis and modeling and data standards, sampling protocols and intercalibration, and program structure and program management and education and capacity building. And so I think this is, you know, here's an example of what we need to come out of this meeting with an outline for, right? And Maite will talk about that in depth. And so I want to reemphasize what Heather said. We're going to reemphasize this all meeting. Think big, but also stay grounded in what's feasible in the near term, right? There's pie in the sky things we could do. We'd love to sample, you know, the moons of Jupiter, but you know, maybe not in the next 10 years. Or maybe that's a different community. Sorry to my colleagues who study aquatic moons. And we want to identify challenges that we can solve. For example, these comparisons and these data management challenges and the logistics challenges, and in this meeting, focusing on the science questions and distilling them down into what can be in this science planning report. And we want people to share their expertise, but also think beyond your domain and aim to build something for the collective good. And a good example of this is looking at the science plan from GeoTraces that was proposed in, you know, the early 2000s and what actually happened, right? And a lot of people have come into this meeting being like, wow, the scope of this sounds big. You know, you're talking about ocean sections and time series and process studies and lab studies, and like, wow, that's a lot. You know, can we do all these things? This is on the left is the, you know, the sketch of what GeoTraces was hoping to do. And you can see a handful, you know, maybe 10 or 12 lines, and the science underlying themes that they were going to try and address, the questions they were trying to answer. And then once GeoTraces got moving, you know, countries found ways to launch projects and connect with the program. And you can see there's many, many more lines over here. We do have these ocean sections, But we also have a lot of process studies that are incorporated, a lot of compliance studies where people found ways to connect their data and follow the standards to participate in the program. And so the end product has ended up being far more than what was originally envisioned, right? And I think that's the think big message, right? Like, it sounds like a lot, but we're asking the whole world to collaborate here. And when we work together, we can actually do a lot. So, some of our hope and expected outcomes from this effort will be to improve our understanding of marine microbial communities and their metabolic function, and to produce tools for data visualization and analysis, to quantify how biogeochemical processes affect ecological hierarchies and the structuring of ocean metabolism at different scales, and to develop and standardize sampling and analytical protocols and bioinformatic pipelines, to manage data across different disciplines, a huge challenge, right? To represent key biological feedbacks within ocean ecosystem models, and last, and most importantly, to train a new generation of scientists worldwide and build capacity in the fields of omics, chemical oceanography, bioinformatics, and modeling, as well as their integration together. So, with that, I thank you, and I also beat the buzzer by a couple minutes, I think. I'm not putting out a lot of work, but thanks very much.
Hello, I'm Maite Maldonado. I'm from the University of British Columbia in Vancouver, Canada. And I want to start really thanking the hundreds of people that have really contributed to the ideas that we are putting forward and the effort of BioGeoScape. And as it has been mentioned, many people have participated in national workshops and meetings. So why are we here? And really, we are here to try to come out with the first draft of the science plan for BioGeoScape. So as you have seen, we have a bunch of national reports to work with that we already have worked with. We also have examples of previous science plans. And here we are to come up with this draft of a science plan. So as it has been mentioned, we need volunteers to contribute to the writing of the science plan. We hope to have it by the end of 2024, but just to give you an example, the GeoTraces science plan took 3 years to be completed. So, this is we have to be patient because we want to really coordinate efforts, and we want to talk to the community and so on. And yeah, at the end, we want to not only have a plan for cruises, but also for lab studies, process studies, whatever we need in order to accomplish our goals. So I just, this is like a zooming in the science plan of GeoTraces. So right now, we don't know how our science plan is going to look like, but we're just using this as a guideline. So when you look at a science plan of an international program, normally what you have is an introduction and overview. You have a proposition of what they want to do and why they want to do it. So this is like the overarching goal of the program. I think we sort of have an overarching goal for our program. We want to look at the ensemble of microbes and how they affect biogeochemical cycles and how they are going to respond and affect global change. And then normally what you have is a series of research themes that are like you're going to work with these themes in order to come up with your research questions, the questions that you want to answer with your program. And often, within research, these big umbrella research themes, you have sub-themes or subtopics. And if you go into a science plan, for example, for a subtopic, they might describe the understanding today, how the field can be advanced, what are the objectives of our program to advance the field in that path, and then what strategic implementation you are going to do in order to achieve your objectives, okay? In science plans, you also have implementation. So how are you going to do this? How are you going to achieve your goals? And in our case, for example, we have to design a field sampling program. We're going to take advantage of technological developments. There will be laboratory studies, incubations, modeling, prediction, intercalibration, and intercomparison and best practice guides are super important, and then bioinformatics. And then also in a science plan, you have a project management, and you have education and capacity building. How are you going to do these things? So for these 3 days and a half that we have together, we are going to focus on the blue, okay? So we are really going to focus on the research questions. This is like the core of the program, and we are going to do this on Monday and Tuesday. And then on Wednesday, we are going to look at these three topics of implementation. And these two we are going to leave because, as you've seen, there is already effort leading those. So we are going to leave those for outside of this workshop. So what I'm going to do now is give you an idea of how we came up with these three research themes that you already have seen in the emails that were sent to you. And in essence, what we did is, because we had all these national workshop reports, we had asked these nations to answer three questions. We asked them to tell us how their nation could contribute to the BioGeoScape effort, if there were any impediments to be part of BioGeoScape, and how they thought they could be mitigated by international collaboration. And we also told them to tell us about the research priorities of their nation. And then what I'm going to show you is the summary for this. So in this map, we have the nations that already had a national workshop or had participated in a pan-European, for example, workshop. So in red is national workshops in various countries. In pink is pan-European workshop in Croatia, and the dash is countries that both were participants on the pan-European workshop, but also had their own national workshop. So in total, we have 31 nations participating in these kind of meetings or workshops one way or another. And there was also the Royal Society meeting in September of last year. So as I said, we asked these nations to answer a series of questions and send us a report. It's just 2 pages report per nation. So this is a summary of question 2, where we asked them to tell us how will your nation best contribute to BioGeoScape efforts. And what we have here is a map of cruises that different nations are already planning, or they go there all the time. We also have process studies that are in place, and we also have the red dots of time series in these different countries. And what I think you can appreciate, there is a lot of effort already to sample the ocean and to understand the ocean. And there is some areas that we have to make better efforts to cover, but we're doing pretty well. So this is very exciting. In this question also, they told us about things like observatories that are in place that could be part of BioGeoScape, technologies, experimental studies that they undertake, infrastructure, and modeling. So I'm just going to highlight some. So for example, there is some marine observatories that we could tap into, like the Sea Explorer or Sea Cycler, the biogeochemical Argo float, so you know about new technologies that Mark mentioned. For infrastructure, there is cool things like the Environmental Genomic Encyclopedia portal. There is microcosm and mesocosm facilities, sedimentary observatories, strain collections, you name it. And for example, modeling, if you look at this box, there is all sorts of models that you will need for these programs, from cell type models to all the way to Earth system models. So I think we are in a very good place to make this work, and the time is right. So for question 4, are there any impediments for your nation to participate in the program? I just made a word cloud of the answers. So these are the things that people came up with. We really need to intercompare and intercalibrate, very important for everybody. Many nations were interested in bioinformatic pipelines. They argued that they really wanted to have best practice guides for sample collection and sample processing and so on. And this is very interesting. Many nations said, we need a common interdisciplinary language. So this is something that I think all of us have to work better at. Like, we need to talk to each other and to really have this common language so that we can really do this program. So for question 3, this is the research questions that are of interest to your nation. We distill them into these 3 overarching themes that we're going to deal with this week, okay? So I'm going to go through these 3 one by one in the next slide. So the first theme that all the nations seem to be interested in is to characterize spatial and temporal distributions of microbes that support key biogeochemical functions by linking their molecular and physiological properties to changing environmental conditions. So in essence, for me, theme one is we want to see who's there and what they are doing and how the environment is affecting them and how they are affecting the environment, okay? So we are going to focus on this theme today. We have come up with these 6 breakout groups. You know, you can think about breakout groups, topics, sub-themes. You know, after this meeting, maybe some of these are going to collapse into a single sub-theme. This is just a preliminary way to organize ourselves. So we are going to break up into these different groups. You know about this because you sign up for them. And what we are asking every breakout group is to answer these 3 questions over here. So what science questions need to be addressed to enable the delivery of the theme goal right here? What are the key science gaps that remain, potentially due to lack of coordination in sampling, experimentation, and modeling? And among these science gaps, which ones are really ripe to be addressed by this BioGeoScape program in the next 10 years? For theme 2, we have, we would like to quantify the cumulative influence of microbial processes on the key biogeochemical cycles and their sensitivity to change. And we are going to do this in day 1 and 2. These are, again, the breakout groups we have come up with. I'm not going to read them. You already know them. And again, we are going to focus on the 3 questions that I just mentioned. For theme 3, we think that we would like to assess the capacity of microbes to adjust to global change and modify environmental conditions in order to improve predictions of the impact on biogeochemical cycles and ecosystem function. We're going to do this tomorrow. And again, these are the breakout groups we came up with and the same 3 questions. So yeah, so yes, it's a summary. We are going to do this research really oriented, research questions oriented meeting for Monday and Tuesday, and then on Wednesday, we will look at implementation strategies, and I will introduce that then. So what I want to charge you with, this is a similar slide with, of what we saw from Heather and Mac. So we want you to think big, stay grounded, think about what is feasible in about 10 years. Science plans are like 10 years ahead. Identify questions that can be answered and challenges that can be solved. As Max said, share your expertise, but think also outside the domain. And don't be afraid to challenge yourself or your nation, right? Capacity building, we are here to also do international capacity building. So maybe your nation is not doing something now, but maybe 3 years from now they will be doing it, okay? And this happen in GeoTraces as well. And we really are aiming to build something for the collective good that is really, truly in this interdisciplinary. So don't be afraid to challenge yourself. And I would like to thank you, and you can scan it if you want to contribute to the science plan. Right.
All right, we have some time for open discussion, and then
I have a question concerning, well, when I read the paper from 2019, there was a strong reference to the seascapes concept. What's the role of that at the moment? So that wasn't clear to me from your presentation, actually.
Did I answer that? I think one thing we discussed as the Science Planning Organizing Committee was the vision statement. And one of the questions that was asked to the nations was, what do you think of the 2019 vision statement? And a lot of people came back and thought that the language was a little bit confusing, or weren't familiar with the hierarchical seascape terminology. So I think in terms of semantics, I think we may adjust that. But I think the concept is still of interest. Would anyone like to add more to that? Yeah. The 4D ocean concept, yeah. Yeah, that's a good point, though. And I think, you know, it's amazing how difficult it is to write a one-sentence vision statement.
But from the national report, there are a lot of nations interested in time series. So I think we'll get to this 4D. So I think we have a chance to do the geoscape.
Thank you, Ingrid.
Maybe say who you are, too, so people online can know.
Yeah, this is Mustafa Yusuf from Meteo IMS, Turkey. So my question is about the national level consultations, which we couldn't do yet one in Turkey, but are we going to have further chances? Maybe once the draft is out, are we going to be able to bring it down to our national level and continue the consultation? So I think maybe this question is relevant for other countries as well.
Yeah, that's very much the hope, right? I think the science plan is a document that, you know, we can bring to our nations, our scientific community, and our funding agencies, and say, here is an exciting effort that the global community wants to move forward on, and we want to figure out how in our nation we can participate and/or, you know, support this science, right? So I think that's very much the aim. It sort of it crystallizes the intentions of this and formalizes it in a way that we can communicate it better to our own nations.
And the reason why these science plans take 2 to 3 years to be completed is because of the consultation process. So we cannot skip that. So that's.
Hi, I'm Federico from Buenos Aires, Argentina. And so for curiosity about the geotraces, while you were planning and writing the plan, etc., you were already starting with like incorporating some cruises or data into that. I would like to know a little bit about that to better imagine the process here.
Would a geotraces person like to respond to that? I'm one of many, but it's more voices talking. In geotraces, there were cruises ahead of time of the program, and how did those get incorporated? Say your name.
It's Alessandro from University of Liverpool. The question was about how do we incorporate the cruises and activity that is going on now into a program that has not been started. Okay, so I think geotraces is a good example here in that there was a lot of field activity that occurred as part of the International Polar Year in 2007-8 in the Arctic and in the Southern Ocean that occurred before the official launch of the program, but was ultimately integrated into the data products and things like that because the data was intercalibrated. This goes back to Mac's point of intercalibration. So I think you can think of these as kind of pioneer biogeoscape studies, proto-biogeoscape studies that are going on now that are addressing these kind of science goals. And the pathway ultimately for them to be included into the knowledge generation and data products of the program is via intercalibration, right? By showing that the data that has been collected on those cruises can be made interoperable, intercomparable with the data that is being occurred in the program. So I think absolutely there has to be a way for the science that is going on now and has occurred before this date to be integrated into the program activities. I hope that answers the question.
Do you want? I can't hear you.
Any questions from the virtual participants?
Hi, so nice to be here. Really exciting to see. I sort of left off on this I think, with the Geomics cruise out of Seattle, and we all got together and had a lot of, oh, I'm super sorry, Alex Warden, MBL, Chicago. But I was wondering at that time, and I guess still, and having watched geotraces and gone on cruises with them and tried to do biological sampling at the same time, I'm wondering how far the conversation has come and how much we need to talk about, or if this is too in the weeds, about the differences in scales of sampling and the 4D resolution, the time factor for offsets of biology from chemistry, et cetera.
Yeah, that's a key question. And I think one that, you know, we can discuss in the breakout groups and throughout the week. But I think to sort of summarize, I mean, I think it may depend on the kind of science questions we want to answer. You know, are there basin scale questions we can answer that don't need high resolution temporal sampling? Are there more specific biological and ecosystem questions or chemical questions even that require high resolution sampling, temporal sampling, and can we combine those within a similar program? Can the data handling systems handle that kind of variation in time and space sampling, and how do we build those pipelines? I think those are great questions. Anyone else like to think about that or talk about that topic?
Sorry. Hi, Ben Twining. Mac, just to add to that, I mean, I think maybe the simplest answer is also just that they haven't been decided yet. And so you haven't missed anything, but this is this is one of the number of places where we're really going to dig into them. And I guess my other perspective to add to the previous question of sort of like, when does BioGeoScape start? And, has it already started or is it starting in some future date? I mean, in the organizing group, I often thinking about sort of the funnel, and it's not like the door closes in the planning process, but the funnel does sort of gradually, narrow things down. And I think we feel like we're in the point where we need to really start digging in on those sampling design and connecting the sampling to the question being asked. And I think that's a real key part of this week. Would you agree with that? Official organizer people?
Yes.
Wanted to put it in another set of words. Thanks.
Yeah, thanks, Ben.
Hello, Damien from Nantes University. I just have a question. Are we thinking having only one system to study, or are we designing the connection between two systems that have been already, like biogeochemistry on one side and more omics on the other? Because to me, it's not very clear from the presentation from this morning if you all are targeting one single system or two systems and how they are talking to each other. And the way we present the whole package is not clear to me, and I think it's important for the breakout session, like if you need to design something new or if you need just to interoperate these two systems.
Just to clarify, what do you mean by system? Are you talking about a scientific
maybe
system?
The question we need to assess together, but
sorry, say again?
It depends. But by systems, you could mean that especially you present the fact that you want to predict. So sometimes if you want to predict, you could assume that you put away a lot of details, or you want to integrate all the knowledge and maybe to design new units of variables that are common between biogeochemistry and omics. So I don't know if it's you could have biogeochemical systems on one side, omics systems on the other, so more or less biological systems on the other. We know that they are talking to each other, for sure. But do we want to increase this cross-talking between the systems, or do we want to build up a new system that incorporates the whole in a way that is not just two distributed systems that are talking to each other? Like in biology in general, this is two different approaches. Like we want to be more holistic or more mechanistic and cross-talking between mechanistic systems.
Yeah, that's a great question. Does anybody want to have a stab at answering that? No, it's great. No, I think it's really key. Want this mic?
Okay, Damien, it's Alessandro. It's a very thoughtful question, as usual. So you're asking. I just want to make sure I understand the question correctly. You're asking about whether, we have capability, we have ways of observing the biogeochemistry of the ocean today. We have ways and capacity for observing the biology, let's say today. And your question, I guess, is about should we sort of continue doing what we are doing, but trying to do it at the same time and in the same places so that we can maximize the synergy? Or should we rethink how we are actually observing this coupled system and thinking about whether the tools we have for observing that are, in a way, set up within those disciplinary silos, and so they kind of, in a way, they impede this crosstalk? Have I understood your question correctly? So I think I don't have a good answer for the question, but I want everyone to think about it because I think it's a really good question, because we're trying to think ahead over a sort of decadal scale, and there may be ways we're not, the way we're observing may be impeding that integration, which then impedes the modeling and the predictability of the system. So I think it's a good thing to get in everybody's mind and to think about in the discussions ahead. I don't want to open a door or close a door in the answer to that question.
Yeah, just to add to that, you can pass that. I agree, it's a great question, and you can almost bring it back to how we teach the science too, right? We're currently largely siloed in our disciplines, I think, in many academic institutions, and do we need to start rethinking the way we educate people in order to rise to the scientific challenge? That's beyond the scope of maybe what this meeting will do, but I think it's something that each of us at our own institutions may need to start thinking about as well, right?
Thanks. Stephen Hallen from the University of British Columbia. And that is an important fundamental question, actually. It gets to the heart of the philosophical dimensions of what underlies the truth and what we're here to try to tell. And I know from experiences with a SCORE working group that looked at microbial community responses to ocean deoxygenation, like we're trying to answer this question in a very narrow way within expanding oxygen minimum zones. And it was about bringing the biogeochemical measurements, rate measurements in particular, into harmony with the genomic representations of microbial life. And one of the things that we had to address was exactly this issue. We didn't resolve it, but we did know that just the way we sample changes the results that we see. And one of the take-home messages from that experience in the SCORE working group was that we need to be doing more in situ types of work. And that could address that harmonization problem if we're collecting the data from the communities in situ and the rates in situ. We're maybe more likely to resemble the truth and improve the modeling efforts.
Thank you. My name is Kai Ziervogel from the University of New Hampshire, and I have a logistical question, actually, before we get into the science discussions. I'm not too familiar with the GeoTracers program, so that's why I'm just asking this question. I'm more familiar with NASA field programs. So there will be a science plan for biogeoscapes in the near future, hopefully. Which will include all the international collaborators. So there's going to be one document. That's a question. And then with that document, the community is going to their national funding agencies to obtain funding for field work to study this. Question number 2.
So the first question, what was the first question? Yeah, one science plan, but there is multiple versions. So after this meeting, we will draft the first one, and then we send it around. The nations read it. They say, oh, no, my nations can contribute this other way, or, so it's really a live document for a couple of years, probably. And then once it's completed, then that's normally what you do. You go to your nation and you say, we are part of this program. We want to be proud of this program. So we want to go to the Arctic. So you write a proposal and you go to the Arctic. So every nation is going to find ways to fund the science to be part of the Biogeoscape program. But, maybe you are a nation that you really have research vessels and you can go all over. Well, you might be a nation that really don't have a field program, and you're going to do a lot of lab studies, or, So I envision this program as multiple ways to be part of it, and, as long as you contribute to the research questions that we want to answer. So it's not very prescribed, Anyway, this is what's going to come out of this.
I want to add one point to that, because it comes back in a way to Fede's question. It's about actually in the drafting of this science plan, part of it will be drafting the way in which science that's going on gets integrated into the program, right? Because in many nations, there is not going to be I can speak for the UK, for example. There's no way that funders are going to say, here's so many millions of pounds to do biogeoscape science. We're going to still fund our science in the way we fund our science today. We're going to write proposals to look at knowledge gaps, blah, blah, blah. And many of you are going to do the same in your nations. The key is, what's the pathway for that science and that knowledge and data that you generate to be integrated into a program that hopefully is greater than the sum of the parts, right? So that's very important, I think, when we're having these conversations, is to think about what's the added value of integrating all these activities that we could go and write a proposal tomorrow to address some of these things. And in many nations, there is not going to, our funders are not going to read a lovely science plan and say, oh, this is great, here's 20 million pounds to go and do the science. But this is something we need to think about as we develop this draft science plan. What are the kind of, when it comes to what Ben was sort of saying as well, it's kind of sharpening that funnel and deciding what is a biogeoscape study, what is not a biogeoscape study. And that can be applied retroactively and going forward. So I think it's a good question, but it's, hopefully I helped a little bit with that answer. And it's for us, the community, to decide where those guardrails are.
Yeah, and I'll add to that too. I think, there is no one international body that can fund this, right? So and this is the case for GeoTrace and other large programs too, right? And so it sort of goes down to the scientists to to consider their own nations and their funding schemes and/or collaborations to participate in other countries that can become vehicles to piece together this program. And then one lesson learned from GeoTrace is that countries had to justify participation in GeoTraces in very different ways. Some countries had to come in with very sort of hypothesis-driven process cruises, and some countries could come in with more, section transit-oriented cruises, and all of that was welcome, and and it had to be tweaked, I think, in order to allow the scientists in each nation to figure out how to generate the support they needed. I did learn recently, though, that there is a new program at NSF called Global Centers that does actually fund internationally between reciprocal nations. There's only a few reciprocal nations right now, and it may depend on what kind of call they put out, but there actually are vehicles to have multinational single grant entities between 2, 3, 4 nations or something like that. And people online, feel free to chime in. We can see all your names on the screen and a few of your faces, so we'd love to hear from people online too. Oh, and I have an important announcement that I forgot to make too. We know we're an official program now because we had stickers made. So I'll put these out.
Hi, this is Noelle Held from ETH Zurich and soon the University of Southern California. So on the topic of motivation and sort of presenting this to everyone's countries for funding, to what extent do we want to involve like policy or policymakers or people who work in policy research in the development of these plans? My understanding is that in the past, this isn't usually included in these science plans, but the world has changed, science has changed. So just wondering if that's been on the radar or if we're really just talking about basic science.
That's a great question.
For the question, Noelle, it's Alessandro. I think you're absolutely right. The way in which we motivate the science we do is completely different now, maybe, than it was when GeoTrace's science plan or SOLAS science plan was written. So I think it's something we want to think about including. Equally, I think something that's very important when we're drafting the science plan is going through that iteration phase, is that we get feedback from those kind of communities. We're all here because we're interested in the science. So, of course, we're very enthusiastic about the science that's going to be conducted as part of Biogeoscape. But once it's out there in the wild, our proposals are going to be competing against lots of other programs and activities who think their program is really important as well. And I know that when the Geotraces Science Plan was developed, there were a group of people who were brought in to the steering committee or the writing committee to bring that outside perspective, physical oceanographers, climate scientists, paleoceanographers, to ensure that the science plan wasn't too, how can I say, siloed within a community. And I think that's something we need to think about doing, and maybe adding to that the policy component, as you say, not just think about other scientific disciplines, but some of the users of the knowledge that we generate. If we want to influence IPCC reports, efforts going on around carbon dioxide removal strategies, et cetera, these aren't really just scientific endeavors anymore, right? And so it's about that science to policy transition. So I think it's a really good point.
I just want to jump in for a sec. I'm Naomi Levine, and we actually have 3 of our Biogeoscapes fellows here. So can you guys stand up, Mariel, Scott, and Ariana? And if any of you guys are online, I'm so excited about this program. So these are the faces. Mariel came all the way from Denmark at the last minute. We just had the program introduced. So, and we're hoping that they're going to be leaders and ambassadors for their early careers. So if you have postdocs in your group or grad students, point them to these guys. And we're really, and we've got, who's online? We've got some, do you want to introduce yourselves online? I can't read on this screen, I'm sorry. If there's any other fellows online. And then, do you want me to?
I can do it.
Okay, yeah.
Are you done?
So anyways, early career fellows, and we're going to have another cohort next year and the year after. So send postdocs, and we're hoping this will be a way to get young people engaged. Oh, Emil, sorry, is that you? Hey, there we go, awesome.
Great, thank you, Naomi. And we also wanted to introduce Leslie Arbach, who is our potential future coordinator for the Biogeoscapes program. If you could give a wave there online. Yeah, she comes with lots of experience from a Bangladesh program and is situated in Tampa, and we're really excited to have her join our team.